Hello, welcome to Module 3, the biology of COVID-19. My name is Alyosha Taramchich. I am a Dairy Extension Associate at Cornell University, and in this module, we will look at some of the characteristics of the virus and infection that will help you understand why it is extremely unlikely to get COVID-19 from food or food packaging, why SARS-CoV-2, the virus that causes COVID-19, cannot survive in food, on food packaging, and other surfaces for long, and why getting a vaccine is so important to protect employees, the work environment, and stop the pandemic. We will first look at what is a virus and how it functions. We will look at the general virus structure as well as specific structure of coronaviruses and how this structure determines the survival of viruses outside of the host. We will look at what is required for a successful infection to occur and how the virus spreads, as well as how our immune system protects us from infection and the positive role vaccines have in this process. Let's start from the beginning and see what virus actually is and how it functions. If you look into all of the biological agents that can cause illness, you will find 5 major groups: bacteria like Salmonella, fungi that include yeasts and molds, protozoa, parasites, and finally viruses. For example, HIV, flu, common cold, and SARS-CoV-2. Now you probably notice that the viruses are separated from the rest. And this is because the first group are actually living organisms, living cells or groups of cells that form an organism capable of growth. And in order to grow and replicate, they need food and they need to have a metabolic activity. Virus, on the other hand, has no cell, has no metabolic activity, and does not need food. It is not a living organism. A virus is a particle or a vessel that contains genetic material. This genetic material represents a blueprint for replication and making new viruses. The way virus is replicating is by hijacking a living cell. For example, SARS-CoV-2 can hijack a cell in our nose. The virus hijacks a cell by inserting its genetic material into the cell and then using the cell's own machinery to read the blueprint in this genetic material and make hundreds of new virus particles that can go and infect other cells and other hosts. This entire process cannot occur outside of the host, so the number of viruses in the environment can only be reduced. It cannot be increased because they cannot replicate in the environment. Now let's talk about the virus structure. As you can see, we have a lot of different virus structures, but all of them can be separated into 2 main groups that also strongly define their stability. The first group are viruses that have the outside part of the vessel made out of protein, and the second group are viruses that have this outside part made out of fat. The fat layer is also called an envelope, so we call the first group non-enveloped viruses and the second group enveloped viruses. Now, if we list the most common foodborne viruses like norovirus and hepatitis A, as well as other viruses that were at one point or another reported to be transmitted through food, you will notice that every single one of them belongs to the group of non-enveloped viruses. A large factor in being able to survive in food is this vessel made out of protein, which is much more stable than the one made out of fat, which means it is more resistant to environmental conditions. Now, if we add SARS-CoV-2 virus, you will see that this one is an enveloped virus, similarly as flu and HIV virus, none of which are foodborne viruses. Enveloped viruses are very unlikely to be foodborne because they are highly unstable outside of the host. The fat envelope is even more unstable if the environment is acidic, and we know most foods are acidic. Our stomach is also an acidic environment, so even if we were to ingest the virus in some unlikely scenario, the virus would not survive the transition through stomach. The fat layer is also sensitive to detergents. If you think about it, we use soap to emulsify and remove fat from our hands, and the same happens with fat layer on the virus. This is why regular hand washing and use of hand sanitizers is so effective in protecting us from COVID-19. And it is also one of the reasons why this virus will not survive in food processing environment where surfaces are cleaned and sanitized on a regular basis. A common question is, how long will a virus survive on surfaces or in food? And the answer is it depends. It depends first on how stable the virus is on the surface. Is it enveloped? Is it non-enveloped virus? How fast it will be inactivated. And secondly, how much of the virus is deposited on the surface in the first place? Or what is the initial number of viruses that contaminated the food? Inactivation is not instantaneous. As you can see in this figure, at the beginning, the number of viruses is high, and as the time passes, the numbers are reduced until all of the present viruses are inactivated. We have two examples here. First one in brown is inactivated fast, but a lot of it was initially deposited on the surface or in food. And the second one in dark gray is inactivated slow, but not a lot of it is deposited on the surface at the beginning. And it looks like in both examples, all viruses are inactivated at the same time. If the virus in dark gray was deposited at the same concentration as the brown one, it would take much longer for all of it to be inactivated. This is something we would expect from a foodborne virus like norovirus: high contamination, slow inactivation. The opposite scenario would be where the virus that is inactivated fast is also deposited at low numbers. It would be inactivated much faster, and this fast inactivation with low initial number of viruses is what we expect from a virus like SARS-CoV-2. Unless we are in a hospital environment where they're performing a lot of invasive procedures that generate a lot of respiratory droplets, we don't expect a high contamination, and we already said that this virus is not very stable outside of the host and is inactivated very fast in the environment. When testing stability of the virus in laboratory environment, high numbers of virus have to be deposited on the surface or used to contaminate the food in order to be able to determine the dynamics of inactivation. These high numbers do not reflect the numbers in real-life contamination events, which are generally expected to be very low. So when reading these reports, look for information about half-life. This would be the time needed to inactivate half of the virus that was deposited on the surface. And this will allow you to determine how stable the virus is. As an example, half-life of foodborne virus like norovirus, which is non-enveloped virus, is measured in days, while half-life of SARS-CoV-2, an enveloped virus, is measured in hours. For example, one scientific report is saying that half-life in water droplets is only 1 hour. Remember that once a virus is inactivated, it leaves behind its genetic material, which can remain present in the environment for a long time and be very effectively detected with modern PCR and other molecular methods. However, this genetic material is not something that can infect you or cause COVID-19. Next, we will look into how viruses spread. Modes of transmission can be separated into direct and indirect. Under direct transmission, it can be actual direct contact like skin to skin, kissing, sexual intercourse, or droplet spread when you breathe, speak, sneeze, or cough. The reason why this droplet spread is considered direct transmission is because the person needs to be close to the infectious person in order to be in range of those droplets and get infected. This would be the main mode of transmission for respiratory viruses, including SARS-CoV-2. SARS-CoV-2 is not typically transmitted indirectly. There is a possibility of airborne transmission through small respiratory droplets that float in the air, but the key is that there has to be a lot of these droplets with the virus present in the air. As we said, a hospital where COVID-19 patients are treated with invasive procedures might be an environment where this type of transmission can occur. But this type of transmission is considered to be rare in other real-life scenarios. What you will not see with COVID-19 is that the person gets infected when entering a room hours after the infectious person has already left the room, which is something we can typically see with true airborne viruses like measles and chickenpox. When it comes to transmission of SARS-CoV-2 through contaminated objects and surfaces, it is possible, but it is less likely. As we said, the virus is not very stable, the contaminations are not very high, and also the transfer rates are very low. Going from hands to surfaces, from surfaces to hands again, and from there to your face, eyes, nose, and mouth, in each step only about 1% of the virus gets transferred, while 99% of the virus remains attached to the surfaces, which results in really small number of viruses reaching your face. This is also why washing your hands and not touching your face is so important, as well as why cleaning and disinfecting frequently touched surfaces should be done as an extra precaution, but only after we have social distancing, face coverings, and hand washing implemented consistently. Cleaning and disinfecting frequently touched surfaces should not be viewed as the main mitigation strategy. We said contamination has to be transferred to nose, eyes, or mouth. You will not get infected through your skin if you have the virus on your hands. Nose, eyes, and mouth are portals of entry. This is how the virus enters the cell, enters the body, and infects a person. In case of SARS-CoV-2, the portals of entry are also portals of exit. So the fluids from nose, mouth, and eyes of an infectious person are the ones that carry the virus that can infect others. Now, to explain why mucous membrane in your nose, mouth, and under your eyelids acts as a portal of entry, we have to explain the concept of receptor. A virus cannot enter any cell. It has to attach to it first, and this attachment is very specifically determined by a protein on the surface of the cell that matches a protein on the surface of the virus. It's like a lock and a key. If it doesn't match, then there's no attachment and there's no entry. The red parts on this model of SARS-CoV-2 virus, called the spike proteins, act as a key, and they match the lock on the cells of nose, eyes, mouth, which allows the virus to enter the body. This need to find the receptor to enter the body is just one obstacle of many that virus has to overcome in order to cause the final illness. Therefore, being exposed to a small number of viral particles is very unlikely to result in illness. The number of viral particles needs to be at infectious dose in order to make illness development a likely scenario. Currently, we still don't have scientific data on infectious dose for this virus, only estimations. Estimations are that the dose is somewhere between SARS-CoV-1 virus and flu virus. One study is reporting that up to 800 virus particles are needed to have half of the exposed people develop COVID-19. Based on this, you can see that different people are differently susceptible to the virus. Part of it is age, immunity, and health status that makes some people high risk, but there are also unexplained factors that make young, healthy people develop severe illness. Factors like type of portal of entry, virus dose, and virus variant determine how likely the infection will occur and how severe infection or illness will be. With this, we will look into immunity, how our body protects us from infection, and how vaccines can help in this process. There are two types of immunity, and each has its role in protecting us from the virus. First line of defense is innate immunity. There is a more general type of immunity that either collects and inactivates free virus in the body, or it kills infected cells because they don't behave as normal cells. This type of immunity is very active when we are young, but as we get older, it becomes slower and less effective, which in part can explain why we see more severe cases of COVID-19 in older population. Innate immunity is rarely enough to completely stop the infection. This is why a week after initial infection, a second more specific immune response is initiated, and we call this adaptive immunity. Antibodies are the main part of adaptive immunity, and besides helping with the current infection, they remain for some time in the body to protect you from the next infection with the same virus in the future. Now, these antibodies and adaptive immunity can also be developed through vaccination. Vaccine introduces inactivated virus or only part of the virus into our body and initiates immune reaction without going through the full course of illness. Only mild side effects are expected after receiving a vaccine, which is a normal reaction when our immune system is activated. Current scientific information shows that vaccination is establishing a stronger adaptive immunity compared to immunity established through natural infection with the virus, which is the reason why vaccination is strongly recommended even if a person already recovered from COVID-19. When vaccines are administered to a sufficiently large part of the population, some estimates are 80% or more of the population, then we achieve something called herd immunity, which refers to low risk of infection for people that could not receive the vaccine. Low risk comes from everyone else around them being vaccinated, being immune to the infection, and not acting as the source of the virus. Vaccination and herd immunity are key elements of limiting the spread of the virus and stopping further development of the pandemic. With this, we are finishing this Module 3, the biology of COVID-19. Remember, a virus is not a living organism. It needs a living host cell to survive and replicate, which is the reason why outside of the host, the number of viruses can only be reduced, it cannot be increased. SARS-CoV-2 belongs to a group of enveloped viruses, which are known to be particularly unstable outside of the host and are highly unlikely to be transmitted through food packaging, or contaminated surfaces. It is important to know that a small number of active virus particles is not enough to cause an infection. Sufficiently high number of virus particles needs to reach the right receptors in nose, eyes, and mouth to cause an infection. And finally, vaccination can protect individuals and population from infection and spread of COVID-19. Thank you.